You got video of them like down like this. Oh yeah, you gotta be like Tennessee, I don't know. So yeah, you gotta say. Yeah.
Whoa.
Yeah, that's
Okay, looks like that mic isn't working today, so I'm gonna be just extra loud with my voice. Can we all hear me? Okay. Yeah, this is how my voice actually projects. Okay. Maybe there's a mic from up there. That's cool. Okay. So we've been talking about synapses. So last time we talked through the different parts of a synapse. I can do that. Project a little differently. Maybe I could just put my screen. What are we doing? Okay. Okay, so we talked about the different parts of our synapse. So we talked about the fact that we have a presynaptic neuron before the synapse. We have a synaptic cleft, then we have a postsynaptic neuron. We have communication between the two from the presynaptic to the postsynaptic. This is working by having the presynaptic neuron release neurotransmitters that then bind to receptors on the postsynaptic neuron. Okay. So that is our example of a chemical synapse. This is going to be a pain. Okay. The smaller. Ready? I want you to be able to see the question. Project it like that. You won't. You can? Okay, great. Even on the slideshow mode? Yeah, you can see it? I feel like you see the choices. Okay, but I can read it to you. So we're looking at our figure, and we are trying to figure out whether we have any molecules from the presynaptic neuron actually getting to the postsynaptic neuron. Yeah, exactly. I think the cutoff is gonna.
Does the screen go up? Like a little bit?
I think it's the projector, not the screen. There's still physical space out there.
But I think the screen is.
That's why I made that extra weird sound. Yeah. All right, good. So we're thinking about, is our presynaptic neuron directly connecting to that postsynaptic neuron. Okay. Good. Go through the details here. But no, there aren't any molecules directly going from inside the presynaptic neuron to inside the postsynaptic neuron. I'm asking you this because this is one of the key differences between our electrical synapse and our chemical synapse. So the electrical synapse is the one with the gap junctions, where you have a tube basically running directly cytoplasm to cytoplasm. So in an electrical synapse, you can have an ion or some type of signaling molecule go from presynaptic to postsynaptic, right? We can even have some bidirectional communication because those insides of the cells are basically in direct contact. But in a chemical synapse, we're going to be releasing neurotransmitters into our synaptic cleft, but the neurotransmitters really just come up and touch the postsynaptic neuron. So you can think of them like they're ringing the doorbell, but they're not actually going inside into the postsynaptic neuron. So that's a key thing to notice as we're looking at our synaptic communication. Okay. So we talked about how action potentials propagate. So we know that those are essentially a wave of depolarization. So we're going up to positive 30 as we have all our sodium rushing in. Right, so that's our action potential reaching the axon terminal. We then have our presynaptic neuron in its axon terminal, so that end bit reacts to the action potential. So specifically, we have a voltage-gated calcium channel here, and when it gets that voltage from the action potential, that opens this calcium channel. So the calcium channels weren't involved in propagating the action potential, so moving it down the axon, but now they are involved with actually releasing our neurotransmitter, so doing this communication piece of an axon. Okay. So our calcium channel's open, so that's that little green one with number 2, right? Calcium's coming in, and it's telling these vesicles, these synaptic vesicles, to dock, so attached to the membrane, and to secrete, so to move these neurotransmitters out into our synaptic cleft. Right, so here's where we can see that piece about the neurotransmitters not actually moving physically into the next cell. Right, so if you notice by number 4, right, our neurotransmitter is diffusing and binding to the receptor, but it's not getting pulled in, right? So we're binding to this receptor. We're not moving into the postsynaptic cell. We have just a response of the cell to this binding movement, right? So this binding event. Okay. So that's how we actually send the signal of some type to our postsynaptic cell. It's active binding our neurotransmitter into this receptor. Then we have to clear away the signal, right? If we don't, if we just leave neurotransmitters hanging out in the synaptic cleft forever, then we're going to have that cell, that postsynaptic cell, continue to react and react and react as if that presynaptic neuron is still telling it to do something, right? So we also want to wash away this signal, right? Because otherwise signals become kind of meaningless if they just hang around forever. So that's what the additional steps past this point are about. They're about how do we get this neurotransmitter out so that our signals are meaningful. So we have 2 ways of doing that. So one way we see here in number 6, so some of those neurotransmitters, as they're floating around in the synaptic cleft, I imagine they bind into the receptor and then they pop out. They're not held there for too long. They might next bump into an enzyme here on the postsynaptic cell. So that enzyme could break down our neurotransmitter, and then we would have the parts that we break it down into, these degraded parts, and diffuse out of the synaptic cleft here. Okay? Or we might have essentially a recycling step. So we talked last time, we mentioned that we have this reuptake molecule here on our presynaptic cell. So this reuptake molecule can pull things back in, right? So reuptake is when we take neurotransmitter or parts of neurotransmitter, pull it back into that presynaptic neuron so that we can save those neurotransmitters for later, reuse them some other time, next time we get that. And next time we want. So those are the different kind of steps here. So these two pathways of like how we're getting rid of neurotransmitters, we've numbered them, but they'd really both be happening at once, right? They're kind of two parallel ways we're removing neurotransmitter. Okay. So looking at those steps, I want us to notice, right? Calcium was our trigger for releasing these neurotransmitters, right? So opening our voltage-gated calcium channel is what eventually causes us to dock those vesicles and secrete out the neurotransmitters. So linking this back up to things we've talked about before, how would we know that calcium is going to come into this cell? Right, we saw that that's what happens when the action potential gets to the axon terminal. But on a more fundamental level, why is that happening? Okay. So what's happening here? I guess this would be a passive process, although this would be facilitated, specifically we're going through a channel. But the reason calcium is going in, right, is that our body has set up our concentration gradient such that we have a lot of calcium outside the cell, like sodium, and it's fighting to get into our axon terminal. It's fighting to get into the cell. So we've set up an electrochemical driving force such that there's a really strong force, a strong urge for calcium to move into the cell as soon as we get an opening, right? So we set this up so that as soon as we get that voltage-gated calcium channel opening, we have calcium rushing in. So that's going to be important not only for synapses, but also for when we talk about how muscles function, because those are going to rely a lot on calcium as well. Okay. So we remember sodium wants to come into cells, potassium wants to leave cells. Now we know we got to remember that calcium, like sodium, wants to come into our cells. The synaptic delay. This process takes a little bit of time, right? So it's not as immediate as we would see in an electrical synapse when we can just have ions or other communication molecules moving from cytoplasm to cytoplasm. Right? This is a little bit of a time delay when we're talking with neurotransmitters and chemical synapse going from our presynaptic neuron to the postsynaptic neuron. Okay. It's about half a millisecond to 5 milliseconds, depending. And you might think that because we have a gap here, that it has to do with that space we have to travel, but that's not actually the case. So remember when we talked about our voltage-gated sodium channels and our voltage-gated potassium channels, remember how they had kind of a time delay on them, right? So this time delay is linked a little bit to this calcium entry, right? So it takes a little bit of time for this voltage-gated calcium channel to open. And in particular, it takes time to go grab these vesicles, signal them, tell them to dock onto our membrane of this presynaptic cell and to release the neurotransmitter. So that's really where this gap of time is coming from, is the fact that this stuff going on in the presynaptic neuron takes a while. We got a bunch of different cell parts that have to do stuff in order to get that neurotransmitter out to our postsynaptic neuron. Okay. So that gets us thinking about speed. So we already know that we had a time difference in how electrical synapses work, fast, versus our chemical synapses. We now know that there's a little bit of a delay there. We're going to see a lot more variation on how fast a postsynaptic neuron is going to respond to signaling from that presynaptic neuron. So within chemical synapses, even though they're slower than electrical as a whole, we're going to have fast and slow responses as we're talking to that postsynaptic neuron on the other side of it. So we're going to think about the proteins that we have on that postsynaptic neuron, right? So what we're looking at here, right, so we had our presynaptic neuron. We've just been talking about that. We're now focusing in on our postsynaptic neuron, so the one that comes after, or on this one. Okay. And when we think about the speed of signal transduction here, right, so the speed with which we get a new action potential or potentially forming in this postsynaptic neuron. Right. I'm just going to point out to us, this is the postsynaptic. Okay. We're going to be thinking about the types of receptors we have, right? So this speed is going to be dependent upon what kind of protein is catching that neurotransmitter and how fast does it react, okay? So here's the faster of our two types on the board in front of us. So the faster of our two types is called a channel-linked receptor or an ionotropic receptor. Right, so our ionotropic receptors are the same thing as our channel-linked receptors. So both of these names tell us something about how they work, right? So channel-linked tells us this is opening a channel, right? So we are linking up our neurotransmitter to directly opening a channel, right? And our ionotropic name tells us what goes through that channel, ion, right? So we are directly linked up to a receptor, neurotransmitter binds into a channel that opens up and we have ions go through. Okay. So we can see that in the picture. So we might, in this case, refer to our neurotransmitter as a ligand or ligand. That just means something that binds onto something else, right? So what we see happening there, right, is like in enzymes, we've got this kind of binding site, this little pocket ready to receive a signal from our neurotransmitter. When our neurotransmitter comes in contact, right, that was the trigger to open this channel-linked receptor, this ionotropic receptor. So we're then going to have ions move, right, depending on what ion moves in or what ion moves out through this channel. Will have some type of effect on the membrane potential of this postsynaptic neuron. Okay. But then as soon as this neurotransmitter pops out of that pocket, right, that channel is going to close back up and the signal gets turned on. So what we're going to notice about these channel-linked receptors, so this is going to be a fast response, and it's also going to be pretty fast to turn it off. Our other type of receptor is a G protein coupled receptor or a metabotropic receptor. Same thing. Metabotropic versus ionotropic or channel linked versus G protein coupled are the two ways we might compare and contrast these two types of receptors. Okay. So these are going to be slower than our channel linked or ionotropic receptors. You might see why immediately, like in our synapse, there's a bunch of steps, right? So there's a bunch of cascading reactions that have to happen in order to get this response going, right? So purely just that makes this process a little slower. Okay. So we see that we have some different types of metabotropic receptors within the metabotropic receptors. Some of them might be really, really, really slow. Some of them might be only sort of slow, okay? But we've got some similarities. So what we have happening here, so our neurotransmitter in this case different one than on our ionotropic receptors. It's this orangish red. So our neurotransmitter has a binding point on our metabotropic receptor. So like in the ionotropic receptor, we have our neurotransmitter fitting into the one. So far, so good. And what we're going to notice, though, about that metabotropic receptor is not, right? So we don't have the ability to just rip that protein in half when we have this neurotransmitter bind in, right? So we don't have a way yet to actually change our membrane potential. Okay. But ultimately, we have to let ions in or out to change that charge. So how do we get there? That's where this G protein part comes in. So the G protein mentioned in G protein coupled receptor is this conglomeration down here. So this group, a little blue pieces, at the bottom of our metabotropic receptor, is a G protein that has alpha, beta, and gamma parts. Right, so we're basically just labeling them ABC in Greek, right? Okay. So that's our G protein. What happens when our neurotransmitter binds into the top of this metabotropic receptor is that our G protein starts to break up. Okay? So what we see happening is that we have this alpha part scooch on down the membrane, scooched on down over here to this enzyme in image C here, or in this simpler image B that's to the right, we still have that alpha part of the G protein scooching on over. It's just scooching directly to an ion channel here. So, let's stay on this image B. This is the smaller one. All right, so neurotransmitter bound to the receptor. Our G protein got activated. It broke up. This alpha part came down. And that alpha part is going over to this ion channel and opening it up, right? So our metabotropic receptors, or G protein coupled receptors, are more complex than the ionotropic one. But of the two types of metabotropic receptors, this is the simpler thing, right? So we can just open an ion channel with that G protein. So we'd call that direct coupling. So we directly coupled this up with an ion channel somewhere else on the membrane. Here we have the slowest of our responses in image C. All right, we gave that the most space. So that alpha part of the G protein didn't go directly to an ion channel. It instead went to an enzyme. The enzyme makes some second messenger molecules. That second messenger then has other cellular responses, one of which is to come find an ion channel. So again, we eventually get to an ion channel because that's the only way we can actually change the membrane potential. But it took us even more steps to get there. Okay. So identify descriptions of these ionotropic and metabotropic receptors. Make sure we can tease these apart. Building up next towards understanding what's going to happen in our postsynaptic neuron after we get this signal. Let's take a look. Okay, all right, that's fine. We're going to come back to our graded potentials and these postsynaptic neurons, but let's go through this. Okay, So ultimately, our next topic we're going to be talking about are postsynaptic potentials. It can be type of graded potential, specifically in our postsynaptic neuron. And we're going to have to get ions into this cell to change its membrane potential. Let's see what's wrong with these earlier two descriptions of our ionotropic and metabotropic receptors. Okay, so when we think about the potential that's going to happen in our postsynaptic cell, it's going to be a graded potential, and it is going to be the result of ions entering or leaving the cell. That's going to be true for ionotropic receptors, but it's also going to be true for the metabotropic receptors. We just take longer to get there. We need more proteins, more systems involved. When we have those G proteins in the metabotropic receptors, right, it is correct to link up metabotropic receptors and G proteins. The G proteins aren't coming in from anywhere. They're already there. They're already linked up to that receptor. You can think of them as almost part of it, right? They're already inside the cell. They're already touching that receptor. We don't have to move them into the cell from anywhere else. They're an internal response of the cell just to the change of the shape of that receptor when it gets touched by the neurotransmitter on the outside. Okay. All right, so yeah, ionotropic receptors are the result of ions entering or leaving the cells, but so are metabotropic ones. We never have to have G proteins come into the cell because they're already there. And we never see our neurotransmitters entering the cell. They're all in the cleft. They're touching these receptors on the outside, but they're not actually traveling into our postsynaptic neuron. Okay. So we're going to do a sort of little blitz review of our graded potentials, because the next topic, our postsynaptic potentials we're about to talk about, are just a fancy way of talking about graded potentials. So you can just you can put your whole name, you can put initials, put some random letter, don't care. The reason this is coming in as a quiz is so I can ask you more questions in a row, because otherwise they only need 3 per lecture. Right. Let's play along. Oh, we're going to start out making sure we understand what actually is a graded potential. These are going to go kind of fast so that we get through it and can talk about postsynaptic potentials. Right, how do we do? Okay, yeah, so we've got our small change in membrane potential is a graded potential, right? We're contrasting our graded potentials with our action potentials, which were bigger and long range. Okay, so next, when we have our positive charges entering a neuron, what's going to happen? Is it going to be more or less likely to fire an action potential? Hey, great. Yeah. So we know that both our receptor types eventually get us to open channels. Channels are going to be related to our different ions which have charges. So when we have positive charges coming into our neuron, this is going to make it more likely for us to fire off an action potential. Remember, we're trying to get to threshold. We're trying to get from -70 to -55. Okay. If we have a membrane potential that gets less negative, what is the word for that? So for -70, and we start to go up, in a positive direction, that is one of these words. Excellent. Yeah, that's depolarization. So we have positive ions come in, and therefore we're wiping out some of that negative charge. That's our depolarization. So just like sodium coming in the action potential is the depolarization phase of the action potential. Same thing, same type of word when we're thinking graded potentials. So what if we have a positive charge leave the neuron? And that can happen through a channel too. Okay, great. So like if we go from -70 to -85, like we do during that after hyperpolarization at the end of the action potential, which we also know is essentially our relative refractory period, this makes it harder to fire action potential. Right? So if extra positive ions are leaving, we're becoming more negative. We're not likely to fire a new action potential unless we have some really strong stimulus come in and counteract this. Okay. We also talked about how graded potentials relate to each other if we have multiple. What can we do here? Right. A little hint with how I described it. What we want to know about graded potentials is we might have multiple signals, right? Might have multiple stimuli. Now we know maybe that means we have multiple neurotransmitters coming in to a specific postsynaptic neuron, right? So we're going to add up graded potentials to get our total response in our postsynaptic neuron, right? So we can add them up in time, right? If we have one neurotransmitter come into the synaptic cleft, it washes away, then it comes back as another action potential comes down, right? That would be summing temporally, or they can sum spatially. So now we're thinking, okay, so we have two presynaptic neurons talking to the same postsynaptic neuron, right? So we're getting one neurotransmitter from over here, we're getting one neurotransmitter from over there. What do? We add them up in space, right? So that's the summation spatially. But we do have stronger and weaker graded potentials, right? So they're not the same size. Action potentials always spike to +30. Graded potentials are a big question mark. Graded potentials don't always go up, right? We don't always get closer to threshold. Sometimes we get farther away. So we can depolarize or we can hyperpolarize with graded potentials. Okay. And we had words for each of these things. So we have a word for becoming more positive. We also have a word for becoming more negative than our resting membrane potential. Okay, yeah. Going up was depolarization, going down is hyperpolarization. So extra polarized, extra negative. Okay. We had other words we used to describe these when we were linking this up right at the front of the action potential chunk when we were talking about our graded potentials. We had another word for depolarizing graded potentials. Yeah, they're excitatory, right? So if we start to go up, we're getting closer to threshold, more likely to fire off an action potential. This is excitatory. We're getting excited. Okay, great. So I got a couple people who got them all right, and I'm sure people that did great. Go. And I know that we're timed, right? So I know some of you might not have been quick on the draw. Okay. Although they're not forward, but that sets us up for what we're talking about next. All right, so we remember graded potentials as responses to stimuli. Now that we understand about neurotransmitters and receptors, we're going to see that when we're talking about a stimulus, in this context, that stimulus is our neurotransmitter coming in, eventually creating a change in some ion channel that either depolarizes or hyperpolarizes this membrane. So this is simple how this works if we're thinking about our ligand-gated ion channel, our ionotropic receptor. So we'll start there. Okay. So imagine we've got our ionotropic receptor. Right? So it just opens when we have a neurotransmitter bind on. Okay. Exactly what happens to that postsynaptic cell is going to depend on what that channel's for. Right? Okay. So if we have a channel opening for our sodium or potassium, right? We know that those have the same directional setup, right? So both sodium and potassium have electrochemical driving forces that point into the cell, right? So calcium, which is Ca2+, and that 2 actually makes it extra strong, right? And our sodium come into cells, okay? All right, so this is causing depolarization, and it makes it more likely that this postsynaptic neuron is going to fire off its own action potential, which is why we would call these excitatory, right? Because we're going to be more likely to fire off an action potential. And then we're going to describe where this membrane potential is. So we're in the postsynaptic neuron, and we're talking about a membrane potential. So our EPSPs, or what we'll be calling these, excitatory postsynaptic potential, basically depolarization in this postsynaptic neuron that are happening as a reaction to a neurotransmitter that opens sodium and calcium channels. Okay. On the other hand, depolarizations aren't our only possibility, right? So we could have a postsynaptic neuron where what we open is a potassium channel or a chloride channel. Okay. Chloride is going to be a little complicated. We'll have a picture of it later. But it is the case that when we open either of these, we're going to have an inhibitory response, right? So an inhibitory postsynaptic, so IPS potential, because this is a membrane potential. So our IPSPs are inhibitory postsynaptic potential, meaning we're going to have some type of a hyperpolarization. Okay. So in this case, we know calcium is positive, but we know, potassium is positive, and we know it wants to leave the cell, right? So if it has this channel open, leaves the cell, it's taking that positive charge with it, we're getting more negative hyperpolarization. Chloride ions, we kind of stabilize our membrane potential a little bit, but you can think of them for this case. In this little drawing, I put, imagine them going, right? So bringing more negative charges with it. Okay. So these are postsynaptic potential. That's how they would work. We're just looking at the ion channel itself, which is what we would do if we were thinking about an ionotropic receptors. Okay? So postsynaptic potentials are happening in response to a neurotransmitter binding to a receptor. And they're either going to be excitatory, EPSPs, or inhibitory, IPSPs. Specific neurotransmitters tend to have one or the other of these reactions, right? So anytime you have a receptor, it responds to a specific thing, right? So receptors can be designed to bind specific neurotransmitters, right? So think we'll have dopamine receptors. Dopamine is a neurotransmitter. Serotonin receptors. Serotonin is a neurotransmitter. But actually our most common neurotransmitters that we're going to see that are clearly linked up to one or the other of these types of postsynaptic potentials are glutamate and GABA. These are two very common neurotransmitters. Glutamate tends to cause these excitatory postsynaptic potentials. So glutamate is going to tend to cause more firing in that postsynaptic neuron, cause more action potentials to be generated. Our most common inhibitory neurotransmitter is GABA. So GABA kind of quiets stuff down usually. So inhibiting action potentials on that postsynaptic neuron. Okay. Again, that's the reminder, right? So excitatory, we're linking that up with depolarization because we know that we need to depolarize to threshold to fire off a new action potential. So our excitatory synapses, our excitatory neurotransmitters, our excitatory postsynaptic potentials are all about those depolarizations, making stuff more likely to fire, right? You might even be able to describe certain synapses as being excitatory, so tending to turn that next neuron on, or inhibitory, tending to turn it off, right? So inhibitory, we're going down, 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 hyperpolarizing, or stabilizing the membrane, making it just hard to get more positive, harder to reach threshold, all of which makes it hard to fire an action potential. So just like that refractory period in our phases of the action potential, right? This inhibitory response in the cell suppresses action potentials. Okay. So here are some pictures of some excitatory postsynaptic potentials being generated. So our simpler example in our ionotropic receptor, fast response, will be our neurotransmitter binding in, opening a channel. In this case, you see both sodium and potassium moving, but we know that there's a really, really strong force on sodium. So overall, with this receptor, what we'd end up seeing is a little excitation, a little depolarization, right? So this little EPSP up here. So if you're looking at a voltmeter, that's what an EPSP might look like. Okay. We see a slightly bigger one in this second image, same direction, still in EPSP. And we see a more complicated process. So this is a slower response. This is a metabotropic receptor. In addition to being slower, this is longer lasting because of all this messaging inside the cell that lingers for a while, even after the neurotransmitter leaves. But we can see this is another way we might cause an EPSP. In this case, what you want to zoom in on if you're trying to predict what's going to happen here is that this channel is closing, right? So we can get kind of complicated with that, right? If we're just thinking about opening, we're thinking about sodium and calcium. But if we think about a channel we might be able to close, which we could do with a metabotropic receptor, right? We could bind and close this eventually. Right? This is another way we might cause a spike of charge, right? Because any open potassium channel pulls you down, but a closed one is going to pull you up. So we want to focus carefully when we're looking at these synapses and these receptors, and figure out what ion channel is in our picture, what ion channel is described in our description. You want to know which direction does that ion want to move. And then you want to add on, okay, is more coming through that channel, or are we stopping movement through that channel? Those are kind of the steps of things you're looking for as you're trying to figure out those responses. So here are some examples of inhibitory synapses. Okay. So in this case, looking at this ionotropic receptor here, actually two ionotropic receptors, we see neurotransmitter binding in. And here's how it works for potassium. All right, so we opened a potassium channel. This channel is just for potassium, only potassium moves through. Right? Potassium wants to leave the cell, so we see this dip. Right, so this is that hyperpolarization, or now we might know that this is an IPSP, so an inhibitory postsynaptic potential, pulling us down. We can see one of the simpler things that chloride does here, right? So one way we might see chloride interacting in a synapse. We might have a chloride channel opening and letting chloride into the cell. Right, so here we see that IPSP again on our voltmeter. Right, so in that inhibitory hyperpolarizing direction. Okay. We also do have this more complicated stabilizing situation with chloride. So stabilizing would just kind of create a flatter line, right? But that might stay flat even when sodium moves in. There is eventually a picture of that stabilizing action if you're curious. So all of our types of postsynaptic potential, PSPs, either excitatory or inhibitory, are graded potential. The way they're happening is because we have action potentials in our presynaptic neuron coming on down to our axon terminal, firing off that at that synapse, releasing neurotransmitter, right? And we're going to have that neurotransmitter binding to a receptor. Depending on the receptor, depending on the neurotransmitter, it will either open or it will close, right? This is going to either let more of that ion in, to increase the permeability for that ion, or decrease that ion permeability, stop that ion from coming in or out, okay? Which means we're going to have either more or less ion flux. So either we allow more ions to move or we stop the ions moving. We're going to pay attention, right, to which ion it is, which direction it wants to go, and whether it's coming in or out. And ultimately, we're going to get an answer to, is this hyperpolarizing us? Is this making the charge more negative? Or is this depolarizing us? Is this making our charge come closer to that threshold? So we'll stop here for now. We have actually already talked through our excitatory synapses, but we'll come here next time, do a little bit of a refresher, and then start to talk about systems of many neurons working together.